Hi, good afternoon, or I should rather say good morning, and welcome to the ZP Developer Zone webinar. This is particularly for our colleagues really in California. So I'm sharing my screen with you. You can obviously see my camera. I just want to say this is not going to just be a sort of monologue about technologies. I do actually have some technologies in front of me. So I will be talking about a little bit about wearables this afternoon. I will be talking about IVDs. Maybe just touch upon CGM in general, which is continuous glucose monitoring, but I'll also be doing a demonstration as well. With all that said, let me go back to this camera and I'll go forward with the webinar. So we're doing this particular webinar really with our colleagues in California in mind. And I'll go forward. So I'll talk about a little bit about wearables. I mean, it's probably fair to say that with UCSD and UCLA, etc., in California, actually one of the sort of centers of wearable biosensors. Um, I was there for a good 6 years working on a lot of CGM technologies that are now sort of billion-dollar businesses within California. I will talk about screen printed electrodes this afternoon. So I am talking about technologies for helping either academia do its research or, in fact, for academia to translate its research into products. So these will be, let's say, a couple of things that I will be discussing this afternoon. I will also be talking about cloud-enabled potentiostats. So if I just stop sharing this for a second and just switch to, whoops-a-daisy, just switch to this camera for a second, you will see that I do actually have a potentiostat in front of me next to some watches as well. And you'll also see that I have my phone here, and my phone's in Bluetooth connection with this particular potentiostat. And I will be using the Bluetooth control or connection from my phone to control this potentiostat, getting the data back, etc. So that will be something that I will be demonstrating shortly. I only say that because I don't want you to think that this is just some sort of monologue from me to you. There will also be demos included today as well. I'll also be talking about commercialization strategies. I'm sure you're aware, but it's not an outrageous statement to say that a lot of good science and engineering actually gets stuck almost at what we call technology readiness level number 3. If you're not so super aware of what these TRLs mean, these technology readiness levels, then a lot of scientific papers kind of get to TRL 3, but a very kind of robust product that's been on the market for a few years, you would argue that it's at technology readiness level number 9. And really, it's just about time and investment will bring you to technology readiness level number 9. I mean, also because we're talking about California, I think it was the founder of LinkedIn that said, if you're not embarrassed by your first product, then you went to market too late. Now, it's a little bit hard to use that philosophy when you're making medical diagnostics, because you could end up unfortunately injuring people or even killing them. But I think there is a certain truth in that, and we will talk about commercialization this afternoon as well. I'll also talk about the fact that this potentiostat that I'm introducing, this Sense It All platform, it can also be used for education and research. So I think if you're watching this afternoon, then you're one of the first people to kind of really see this potentiostat put it through its paces. So as I say, I'm very interested at ZP, or we are very interested at ZP on IVD, in vitro diagnostics, and also point of need testing. So, markets that are kind of becoming interesting or are interesting are food testing, agriculture, point of care medical diagnostics. And then that's also now becoming wearables as well. I mean, I know at UCLA, there's a big, drive on wearables, but also the same in UCSD as well, and with people like Professor Joseph Wang down at UCSD. I will talk about microneedles. In fact, this is a watch with a microneedle technology on the back of the watch. This watch has had something like 20 million US dollars spent on it so far, and this will be, if only briefly, I will touch upon it. I think what I will say in this webinar is, in fact, we do a webinar specifically on wearables. So if wearables is your thing and you're watching this video later on, hopefully you'll see some links and you can actually come to the wearables webinar. This might be worth knowing about, but I'm going to do a demonstration later on, and that demonstration is going to be what we kind of call sensor to API, sensor to application programming interface. Now, if you want to at least partly do what the demonstration that I'm going to do, you will need what's called a Julie account. There's a URL here, julie.zimmerpeacock.no. When you register for Julie, you'll register with your email and you'll put a password in there. If later on then you follow these demonstrations, the details that you use to register for Julie, if you then use them to log into what we call the Sensi All app. So if you look at my phone now, you will see that I've got an app on my phone. This app is, I've used my Julie credentials to essentially log into this app. This app is in connection with a cloud system called Julie. So sequence of events, register for Julie. Julie is really the only commercial electrochemical or electroanalytical data management system on the market. It's free to open an account. You can then use that account actually in synchronization with a smartphone app called Sensi All. And why you may want to do that will all become apparent pretty shortly. So just FYI, Zimmer and Peacock, this afternoon, I am going to talk about wearables, I'm going to talk about IVDs, I am going to talk about potentiostats. The backbone at Zimmer and Peacock, our technology stack backbone, is called Sensi All. Sensi All partly is on the Google Play Store. So if you use an Android phone, you can scan this QR code and it will install an app called Sensi All. Pre-register for Julie on first of all, because when you can then log into this app using those Julie credentials, and that will then synchronize the Sensi All app with the Julie database. Because we are talking about California here, so if you're near Palo Alto, maybe you're more interested in iPhones, you can also get the same app for your iPhone as well. And I was talking to a couple of professors the other day, and they really appreciated that the demonstration I'm about to do you is really software agnostic. It's all clouds driven. So you're not installing, for example, if I look at some data in a minute on my PC, I will not be installing apps. I'll actually just be using the browser as a portal to online databases. So I just want to show you very quickly. So hopefully you can see my screen and you can see this. What I'm going to do is I'm just going to tap a button, this button on my app, and now is now asking to scan a QR code. I'm just going to scan this QR code here. Understand this now. It says on my phone, amperometry. This this phone is in Bluetooth connection with in fact, I've got the cable a little bit wrapped up here, is in connection with this potentiostat. So now this will do an amperometric experiment. I'll just put the potentiostat down for a second, and I'm just going to do another QR code. Now don't forget, you can actually try this for yourself. Open circuit potentiometry. I'll just scan the QR code. Right, this app is now able to do an amperometric experiment, and it is still connected by Bluetooth to the device. In fact, it even says device connected at the top here. Another bit of the demonstration, I will scan this QR code, one of my favorites. Impedance spectroscopy, very powerful, because actually this, let's say, little potentiostat here, which is in Bluetooth connection, is able to do impedance spectroscopy as well. Please understand that this is not the typical kind of lab instrument that you're otherwise used to. This is something that's much more akin to, I mean, somebody described it as a flip phone, but I do agree that it's actually much more akin to a kind of an Apple type experience than a normal scientific experience. In fact, I've got some potentiostats in the office here, but what you'll notice is there's no trailing cables, etc. I'll just scan this QR code here, and now I'm able to do linear sweep voltammetry. If I scan this QR code here, I'm able to do cyclovoltammetry. So now I'm in my cyclovoltammetry mode. And if I scan this QR code here, I'm in my square wave voltammetry mode. So very new type of technology, very small, very nice form factor. Bluetooth connection with my phone. My phone scans QR codes. My phone tells the potentiostat what to do, and you'll see a bit more of a demonstration of this short while as well. It's probably worth knowing that, well, in fact, there's all the QR codes, let's say. it's probably worth knowing that it's very easy for us to go from electrochemical method. The whole data pipeline is taken care of in the cloud. So what that means is that you can program the potentiostat to do cyclophotometry. You can tell it how to analyze that cyclophotometry. You can tell it what the calibration factor is, and it will give you a real number. So what holds most electrochemical analytical electrochemistry back is the conversion of raw signal into actual meaningful numbers. And I'll be able to show you that all of that is taken care of in the cloud system that I will show you shortly. I'm just going to slightly go into wearable biosensors for a bit, because at least a subsect of this conversation this afternoon is actually about wearable biosensors. So at Zimmer and Peacock, you've got wire sensors that originally, I mean, for example, this is an Abbott Freestyle Libre 2. I'll just hold this up to my camera. This technology originally, there was a company, it came out of Texas University, a guy called Adam Heller, then made its way up to Alameda. You've got Abbott up there. These days, I think most of the work on this is actually done in Ireland. But this is a glucose sensor. It's called the Abbott Freestyle Libre 2. The fine little filament that you can just about see against the end of my finger there is a complete electrochemical glucose sensor. So that's something that kind of goes transcutaneously and measures glucose in the interstitial fluid. Something that's also going on quite a lot in California these days, you've got BioLink, obviously, in part a spin out of Joseph Wang's lab down in UCSD. Now, this is not a BioLink technology, but it does use microneedles. So BioLink and this technology that I have here are both using microneedles. Now, Zimmer and Peacock, we were the biosensor developers for a company called PK Vitality. PK Vitality were also working on, this is a sort of nicer model. They were working on these kind of attractive watches. These are kind of smart watches, but they were using microneedles in order to put the enzymatic reaction through the skin and into the interstitial fluid. So BioLink is very heavily involved in this. I think in the last 4 years, BioLink has raised something like 158 million US dollars. ZP has a different business model. We do contract development, contract manufacturing with four other companies, rather, I should say. So we are actually licensing this technology into other companies. We also work on non-invasive. Again, I keep referencing Joseph Wang, but Joseph Wang a few years ago did a paper on measuring lactate in sweat. We were actually able to repeat that work quite nicely on our commercial platforms as well. And something else that we do a lot of is implanted sensors. So you do have in the glucose space a company called Senseonics. Senseonics has probably spent some hundreds of millions so far making a long-term implantable sensor. The sensing package that we're doing, it sounds a little bit ridiculous, but we're actually implanting into fish. Now, if you are interested in wearables, I've kind of slightly taken away a few slides out of this presentation because I wanted to focus more in vitro diagnostics. But on the 31st of July, you can see a lot more slides and a lot more information about wearable biosensors. What you will find out is that with ZP, whenever we're doing a webinar, we tend to do one in the morning and one in the afternoon. The reason we do that is because we like to cover the various time zones. We primarily sit in Europe, so in fact, it's quite good being in Europe because if we do one in the morning, then we cover Asia, and we do one in the afternoon, then we cover the Americas quite well, including all the way to California, for example. So what I'm going to talk about now is a bit of a Sense It All platform. So Sense It All for us, as I say, means sensor to cloud, and so that means hardware, apps, cloud database system. So you'll see all that in action shortly. So as I say. Now, one of the things that ZP that we do, what I'm going to talk about a lot is actually screen printed electrodes. Now, screen printed electrodes are not a bad form factor for doing R&D upon and eventually going to actual market. In fact, I've got a glucose system in front of me here. You know, the reason that people are kind of quite interested in screen printed electrodes is because they're quite used to commercial systems where people will take a drop of blood. The drop of blood will be quickly analyzed on a meter such as this. and they'll get a result essentially there and then, and the diabetic will know their glucose. So screen printing has proven itself commercially. It's that it can scale, it works, and you can make products out of it. So you've got the One Touch Ultra, which was made by Johnson & Johnson. This is one of the best glucose sensing one of the best glucose sensing technologies on the market for a while. When I say one of the best, it was one of the best selling. You've also got other companies called Radiometer as well. Just as an FYI, Radiometer have a subsidiary in Carlsbad, California called SenseDX, and they actually use a lot of thick film printing. They make biosensors, and they make them, I think, exclusively for Radiometer. But just as a kind of reference to California here, I obviously know what they're doing because I've read their patents. I don't have any other relationship other than that. But it's just to say that screen printed electrodes have proven themselves commercially to be viable. Either glucose strips, or you can use them more broadly than just glucose. That Radiometer do what's called the comprehensive metabolic panel, for example. The main problem with the main problem with screen printed electrodes that are sold into the R&D market is actually price. So what I mean by that is, here's a sheet of screen printed electrodes. They all look pretty good, and people expect these kind of screen printed electrodes at a good price or low price. And at ZP, we do achieve that. But what is not discussed so much actually is people are getting these electrodes at let's say, a low price, but actually they're not very good quality, and that's a bit of a problem. And I'll talk about why that is in a minute. And the reason it's about, and I'll expand on this, is there's actually more choice than there is demand in this world. So what do I mean by more choice than demand? There are more people willing to produce more screen printed electrodes than are actually needed by the R&D market. So you've got a very saturated market, all fighting essentially for crumbs. And it's actually leading to a quality problem. There's also an over-eagerness and an oversupply of what we call custom screen printed electrodes on the market. And I'll talk about, do you need customization? And if you do require customization and you demand a low price, what you're going to end up with is low quality. Low volume, low price equals low quality. I'm afraid about that. So let me talk about this problem with SPEs. What's wrong with screen printed electrodes? So screen printed electrodes do make for good products. Look at the glucose strips. Billions of these have been sold. Tens of billions have been made in both revenue and also profits as well. But what's wrong with the screen printed electrodes that are making it to the market? The thing is, in some industries, people need a high quality and they're willing to pay a high price. So, for example, you've got a defense contractor in California, in La Jolla, called General, I forget what they call General Atomic. And, they will sell drones to the US Army, and those drones will sell for a very high price. Why? Not just the US Army, the US military, I should say. So they sell drones and they sell for a high price. Why are they so expensive? Because the US military require them to work every time. So they require high quality and they're willing to pay a high price for it. Now, if you look at screen printed electrodes, scientists also need a high quality because actually what you're trying to do is make objective measurements, often of concentrations. And so you can't have screen printed electrodes that are highly variable, because when you measure the same concentration on different electrodes, you'll get different signal strengths. And it really confuses the science and holds back the commercialization. So there's a problem that you want high quality, but in fact you can only afford a low price. And actually it really leads to a problem. So at ZP, we're very good at actually achieving the high quality and achieving the low price, but it's only because we have a completely different philosophy to everyone else on the market. And it's because we really understand that to get a high quality and a low price, you really need high volume manufacturing. And this is the problem, that most screen printed electrodes that you would otherwise purchase are not high volume manufactured. So if you look at the market, you'll find that among many, there's Zimmer and Peacock all trying to produce screen printed electrodes and made them available and people are buying them. But actually, if you talk to anyone who's using screen printed electrodes, other than from ZP, they're always complaining about them. Now, the reason being is you could think that there's at least 10 suppliers of screen printed electrodes in the world. Sometimes an academic team will also have a screen printer and they'll be producing their own screen printed electrodes. And what this means is you've got at least 10 people in the world that will supply you with screen printed electrodes. If you go on their website, you'll find at least 50 products. That means that there's probably over 500 products on the market that if you ask the question tomorrow, I need a screen printed electrode in order to start measuring, the caffeine in coffee, for example, or the chlorine in pool water, or the hepatitis B virus. I need a screen printed electrode to do that. You will find lots and lots. In fact, there'll be over 500 products to choose from. But in reality, how many screen printed electrodes experiments are done in academia per annum? I would, back of the envelope calculations, suggest that there's only 1 million at most screen printed electrode experiments done in academia per annum. So you've got 1 million total requirement in the global market, in the R&D market, served by 500 products. There's actually more than 500 products. There are more than 10 vendors. Most of them are carrying more than 50 products. So this is actually much worse than I'm describing it. So what you actually end up with, if this demand is evenly distributed across all of these different products, you're going to end up with actually each product only selling about 2,000. If you're in UCLA or in UCSD, talk to your screen printed or your colleagues that are using these kind of screen printed electrodes and ask them, how many are you using per annum? You will find that there's not that many. And so what I'm saying here is 1 million is required. There's over 500 products. 1 million divided by 500 is 2,000. Now, if I hold this sheet up, there's 300 screen printed electrodes in this sheet alone. This means that I could fulfill the entire market for most products with 7 sheets. This is not high volume manufacturing. This is not even low volume manufacturing. This is barely enough to turn the machine on manufacturing. So there's a very finite demand, an oversupply from both academia and from industry. SPEs are made in very low volume. In fact, they're made, and most of them actually end up on the shelf, where they're basically aging. And so you're not surprised then when you actually can't find that they're very reproducible. It's only high volume that can drive low prices and high quality. Now, at ZP, so what's actually happening is you're paying a low price, but actually you're ending up with the low quality. And I don't need to defend that comment. Just ask around and you'll soon find out that that's actually is a definite experience that most people have. Now, at ZP, we have managed to achieve high quality at a low price, but it's because we have a very different attitude. So when people contact us, okay, sure, we're part of the problem. We actually have lots of screen printed electrode designs, but when I realized what it was doing to the market, I said, okay, if you want a carbon electrode, Zimmer and Peacock will give you one, but we'll suggest the 501 carbon screen printed electrode. If you want platinum, the 501 platinum. If you want a really low cost carbon, this one. If you want a really low cost platinum, this one. If you want a really low cost gold, this one. Understand that most 90%, 95% of people who come to ZP are only buying one of these 5 electrodes. So we're not trying to please or appease everyone by giving them exactly the design they want. We're actually advising them, please don't go for all these designs that we don't sell a lot of, because in fact we don't manufacture them in high volume. We've made these the cheapest ones on our website to both encourage, and I'm encouraging by telling you that because we make high volume manufacturing of these, they're actually the best quality as well. But I'm also going to show you that we do something extra, which is the amount of testing that we do to make sure that they're of high quality. This quality is really important when you're actually trying to commercialize science, and I'll come to that in a minute. But price-wise, we're doing these at like $1.76. These ones we're doing them at like 90 cents each. So we are getting even though we're only selling in pack sizes of like 300, we are actually able to give low pricing. I want to talk about quality now. First of all, there's lots of people, this happened to me, quite a few years ago when people would contact us about screen printed electrode and start asking questions about, cyclic voltammetry. And I understood why, because this is the way people have been trained. Well, tell me about the cycle voltammetry on your screen printed electrode. The question is, don't ask me about the cycle voltammetry on screen printed electrode. The question should be, tell me about the reproducibility of your screen printed electrodes. Because the reason that you're using screen printed electrodes is because you're actually imagining that one day you're going to make a low cost product. And so you're actually more interested in reproducibility than you should be in the cycle voltammetry. Now, I would say at ZP, we are actually making screen printed electrodes that will pass the cycle voltammetry test. That our signal versus square root of scan rate is linear. So it means that we kind of follow the sort of Randall-Savich equation. Our peak-to-peak separation at low scan rates is something like 59 millivolts per electron, which is close to kind of Nernstian thermodynamic reversibility. So, tick. But it's not important. It's useful. And if you're grossly, let's say you've got a massive separation between peak to peak, then you do actually have a problem because you've got high resistance electrodes. But, for example, we get at 10 millivolts, a peak-to-peak separation of 67.5 millivolts. The fact that it's 8.5 millivolts off 59 millivolts, I don't care. It's not what's stopping you get to market. The fixation on screen printed the fixation on cycle voltammetry is what's stopping you get to market. So I want to break that fixation. If you come to a ZP demo, so ZP makes some products that actually are used in the food ingredients testing. If you come to one of our demonstrations ever, or you see them online, first thing you'll notice is no calibration. How many scientific papers in the electroanalytical space where they're using screen printed electrodes, is there either a normalization of data or some sort of calibration routine going on? Or in fact, what's happening in the lab is that a lot of data is being thrown away because it's described as outliers, where really nobody's quite sure what the outlier problem is. But actually, the outlier problem is actually the screen printed electrodes are not reproducible. You come to one of our demos, we do the demos live, they work every time, and there's no calibration. Why is that? Because our assays are simple. Why are assays simple? Our assays are simple because that makes it easier to commercialize. And the only reason that we're able to get away with no calibration, which leads to simpler assays, which means it's easier to commercialize and it becomes impactful, is because the underlying screen printed electrodes are such high quality in terms of reproducibility that we don't need to have all this kind of daft calibration involved in our assays. So let's get to reproducibility. So if we make a sheet of screen printed electrodes like this, we actually do test by cycle voltammetry. So I just made this comment about cycle voltammetry not being as important, let's say, when with screen printed electrodes, but we actually use it because if we can't get a decent cycle voltammogram out of it, then those electrodes are not going to be generally useful for people doing electroanalytical work. So we do test by screen printed by cycle voltammetry, but we're more interested actually in our reproducibility. And by the way, this data is all crunched using Julie. We'll be coming back to Julie in a bit. Julie is the only commercial online database able to receive, process electrochemical data. Anyways, we built it for ourselves and it's also available online as well. So we take sheets of screen printed electrodes and we sacrifice electrodes from the sheet and we test them to make sure that we have reproducibility. And so, for example, we'll take a number of sheets, we'll measure them all with ferricyanide, we'll look at the peak height, make sure it's within expectations. We'll also check the standard deviation and the relative standard deviation. If you want to be a diagnostic that's going to be useful to clinicians or a diagnostic that's going to be useful for water or food quality testing, you need a relative standard deviation of your electrodes of less than 10%. So we always make sure that we're much less than 10% because we know that if we don't get a good reproducibility, the company that's going to be first impacted is Zimmer and Peacock, and then the people who are going to be second impacted after that is actually all our academic colleagues who are also using the screen printed electrodes. What makes Zimmer and Peacock quite unusual as a manufacturer of screen printed electrodes is the biggest consumer of ZP screen printed electrodes is ZP. It's just the biggest difference. We don't make them for academia, we make them for ourselves, and we happen to sell them on the web stores as well. So really important. I'm going to jump onto potentiostats in a minute, but it's very important to understand that actually, what's driving some of the problems in academia and in commercialization in general is actually the underlying screen printed electrodes. If you come to one of our demos or you use our screen printed electrodes, each one of these colors in this particular, let's say, group of cyclophotomegrams, each color actually represents a concentration. And each color, so for example, here, these yellow CVs, those are actually 3 screen printed electrodes. This slightly tanned group of CVs, that's actually 3 screen printed electrodes, all tested at the same concentration. And then the last kind of 3 gray CVs, 3 screen printed electrodes, all tested at the same concentration. And you can see that they actually all give similar peak heights or area under curves. And it's that then that allows us to be able to make assays upon these and make those assays actually into products. Because our error bars due to the screen printed electrode to screen printed electrode variability is small. If you do adopt our screen printed electrodes and you're not using one of our potentiostats, make sure you get connectors from us, because I've had people, we make beautiful screen printed electrodes and sometimes they can look terrible. And it's basically because the connections that they're making to the screen printed electrodes are terrible. The one thing about electroanalytical chemistry, your data is probably only as good as your weakest electrical connection, or if you've got high impedance connections, it can really mess up the electroanalytical data. So if you're not using one of our potentiostats, please make sure you get at least a decent connector from us to be the interface between our screen printed electrodes and your particular potentiostat. We're quite expert on understanding other people's potentiostats, so we can always recommend the right connector for you. Something that happens a lot now, I think I'm starting to get the message through to people, is people are very interested in custom screen printed electrodes. People come to us and say, oh, I need 2 working electrodes. Often when people are saying I need 2 working electrodes, they'll start describing this idea that they've got an enzyme electrode and a non-enzyme electrode, and they'll do a subtraction. That's because they've had problems with screen printed electrodes, and they think, oh, I've got this background signal, I need to subtract the background signal. What I'll do is have 2 working electrodes, one with an enzyme, one without an enzyme, and I'll subtract the 2 signals, and that will give me my signal as opposed to my signal plus background. I understand the theory. In practice, it has been tried by commercial companies. Dexcom did try it on their Generation 3 CGM. I've seen it tried to be used. It's fixing a problem that's due to your manufacturer of screen printed electrodes. It's not an intrinsic problem. It's just you've got a problem of experience of the screen printed electrodes you've used in the past. It's not a good philosophy to try that. We do get requests. Everyone wants to do multiple analytes. If you want to do multiple analytes, it's probably worth talking to ZP about this. We do get requests for unusual designs. Unfortunately, sometimes these requests for unusual designs, at least in companies, they lead to the liquidation or the bankruptcy of the company. The company's going to Chapter 11, and I'll discuss that as well. So be careful. The other thing that we get is interdigitated electrodes. People seem to think that there's something mysterious about interdigitated electrodes, and I believed it as well for years, until actually I got a lot of experience and then realized there's no extra secret value in interdigitated electrodes, and I'll give you an example of that in a minute as well. The true cost when you're going for different electrode geometries is you want them at a low cost. You want them at a low cost because you've been told that glucose strips are made by thick film printing. They're low cost. So you have an expectation that you want them at low cost. You can have low cost, but when you're going for customization, you want them at low cost and you actually want them at low volume. Low cost times low volume, it means low quality. I think ZP is the only people who actually tell you that that's the equation. There have been companies in the Chicago area that have gone into bankruptcy in the last few years, and really they were just feeding the market with low quality screen printed electrodes. So it's a problem when you're asking for customization. You also can end up what's called low yields as well. So the company that was actually going for this design, they went to several vendors. They came to Zimmer and Peacock. This is ironically the case that they said, you're 3 times more expensive than the lowest price person. So they went with the lowest price person. And the lowest price person produced them hundreds, if not thousands, of these screen printed electrodes. They found out that they were very variable. So they ended up hiring people to do quality control on these screen printed electrodes. They found out that, in fact, only 1/3 of the screen printed electrodes were any good. So they paid low price, but they could only use 1/3 of them. So it was kind of ironic that they said we were 3 times more expensive. It's because I was trying to give them 100% yield when the other vendor was just going to give them the screen printed electrodes. They look completely fine. That's the problem with screen printed electrodes. They can look completely fine for visual inspection, but once you start using them, you find out that the electrode surface is different and the sensitivities are all different. So they had a low yield. They hired a lot of people to do quality control. So they ended up paying the true price for this through basically salaries, and they ended up in Chapter 11. So be careful about asking for customization because you'll get it, and when you ask for low price, you'll also get that, but you'll end up with low quality, and it can be as dramatic as actually going out of business. Um, the big secret, and this also refers to interdigitated electrodes as well, is that at ZP we've got rectangular electrodes, we've got spiral electrodes, and we were testing once the conductivity of a liquid using rectangular electrodes and spiral electrodes, and we used a metal Toledo, just normal lab conductivity sensor, or probe, I should say. They all gave the same results. All gave the same results. Very different geometries. The spiral electrode is like a long line. It's like a line electrode. The rectangular electrode is just a rectangular. In fact, it looks like this and like this. The metal Toledo conductivity probe is two platinum electrodes facing one another like this. They all gave the same results. I realize now after some years that actually, when you're talking about aqueous solutions on relatively small not micro, millimeter electrodes where gaps are very small, electrogeometry is not half as important. When you're talking about large electrodes, high current, large distances, I can completely understand how electrogeometry, electrode configuration is really important, but not on the millimeter scale that most people are working on when they're talking about screen printed electrodes. It's just not as important as people try to otherwise make out. Where can you have the most impact? Let's talk about intellectual property. If you're developing electroanalytical sensors or biosensors, where do you add the most value? Is the most value in electronics? Is it in the hardware? No, it's in the bit that's most mysterious to most people, and that's actually the electrochemical assay. So if you've got 10 hours in your life, do you spend 10 hours building new electronics, or do you spend 10 hours building a new bioelectrochemical assay? The real value is in the bioelectrochemical assay. That's the part that not many people in this world can do. Where else should you spend your effort? You should spend your effort on the method. How do I get the best signal out of my electrochemical assay? So a simple way of saying that is, for example, you could test a pharmaceutical drug using cyclovoltammetry. But actually, we all know that you could improve that signal by actually using square wave voltammetry. But you want to optimize that square wave voltammetry and get the best possible signal out of that assay. So spending effort on that method file is good. The really good place also to spend effort is, let me use the pharmaceutical drug analogy. So it's a pharmaceutical drug analogy and you're trying to test in blood. So to actually get hold of some animal blood, spike the drug in that blood, and do a calibration in that blood. I know it's not going to work terribly well because blood's a hard matrix, but working in that matrix and coming up with a routine to get the signal out of that total signal is a good place to spend your time. So that's kind of essentially calibration. These are the three places, if you put effort in, that's where you add the most value. These are the patentable parts. People, if you have an electronics background, you think all the values in electronics. Electronics are becoming very kind of almost off the shelf, but also very commoditized as well, especially in the world of electrochemical assays these days. Let's talk about having impact. So once upon a time, well, not once upon a time, this is true today. You've got technology readiness level number 1, technology readiness level number 9. Now, most academia actually finishes at TRL 3. It means you go into a lab and you get a proof of principle and you produce some raw data and a calibration curve. And so you've proven it to work in the lab. So that's really TRL 3. And you might do it on a potentiostat like this. You might do it on a much smaller potentiostat with cables, etc. But it looks like a lab experiment. Now, to commercialize, you actually want it to be at a much more robust form factor that can be used by most people. I'm very biased. I'll just change my cameras just for a super second. You want to be working on an instrument that looks like this. This is the sensor or potentiostat. I'll just change cameras back again. So we would, in fact, the sensory or potentiostat is much something that you could actually give to real people. And when I say real people, I don't mean there's fake people and real people. I just mean the people who are non-electrochemists, for example, people that, like doctors and nurses. Now, there's a gap between most of the time that you're doing your research and actually something you could generally give to somebody who was not a specialist. And governments have been, in the US, you have SBIR grants. It's all about closing the gap between actually research and productization. So governments have been spending a lot of money over the more than 20 years to close this gap. This is for me, this is a bit of a game changer, because at ZP, what we're actually suggesting is do your development on an instrument that can also be your final product. So suddenly the gap between TRL3 and TRL9 just got a lot smaller, because in fact you're going to end up using the same instrument to actually go to market, at least to achieve what's called an MVP, a minimally viable product or prototype, depending on your definition of the P part there. So I'm going to expand on this a little bit now and just say about the Sensi All. We are very interested in getting this Sensi All platform actually into teaching as well, because I think teaching is not, electroanalytical chemistry is not always taught as an undergraduate subject. And if it is, it's done on potentiostats that kind of are very off-putting and don't inspire people that this is actually a cool science. And it's one of the not easiest is a bit of a term that's a bit strict, but I would say electroanalytical sciences, one of the, at least in my experience, one of the easier sciences to actually commercialize. At least when I saw, at least deep tech sciences. I mean, if you're trying to make a pharmaceutical, you would have to kind of build up $1 billion company. But with electroanalytical science, for example, I've known people who are working on, sensors for pool water, sensors for water quality. You don't always have to raise billions in order to do things like that. So we're very keen to kind of get this potentiostat actually into the undergraduate teaching as well, and I will expand upon that as well. Because we want to give people, when people are doing either their research or doing their learning, experience that's really relevant, is up to date, and it's actually practical. And I think this is what this Sensi All and potentiostat really does achieve it. It can go the whole journey from undergraduate to graduate to actually entrepreneurship, all in one hardware. We really want to do away with the perception at the moment that electroanalytical sciences, electrodes in fairly large volumes run by square boxes where you need a sort of laptop. In fact, you can do most electroanalytical chemistry now on your phone using screen printed electrodes and using a device that, is, fits in the palm of my hand. And this is something that I do want to talk about essentially next. So we want to keep it modern and relevant and super up to date. We want to keep that experience there. Now, in order to follow this along, you may need a Julie account. So if you want to do this for yourself later on, register for Julie. When you've got that, you can also put this app on your phone, log into the app using your Julie credentials. I will send a recording to you, so you don't have to. I should have said this at the very beginning. If you attend today, you definitely get a recording of this. You can scan these QR codes and you can go between all these different methods. This is only useful if you've actually got the hardware, but I just want to let you know that the software is already developed. And I also want to just tell you that it's very easy to go from lab experiment of cyclophotometry, square wave photometry, amperometry, open circuit potentiometry to actual analytes using this platform. And I'll do you a demonstration, in fact, of that in a minute. I also want you to know that in fact, I just want to go back slightly. I just scanned this QR code. So I'm just going to scan a quick QR code. There we are. I've just scanned the QR code. So actually I can do a demo now. I just want to show you this final QR code. This is the kicker, let's say. If you ever scan this QR code and you've got the app on your phone, you will understand that actually what we've built into this is a true platform for you to commercialize your science. I think this goes down particularly well in sort of, in the USA, where there's a natural sort of, entrepreneurship. We have built in the idea that this is a platform that somebody can adopt and go to market much quicker than they would have historically even dreamt of doing. They thought they would have had to build everything. In fact, it's more like developing an app these days, and even the app is developed. So these are all editable fields. So I hope you get the hardware and you'll be able to try this. We are doing an educational starter pack as well. So if people are interested in using this for teaching and inspiring students, then there is definitely a pack. I want to just talk about sensor to cloud. So what I'm going to do now is a quick demonstration. So I'm going to change my camera for a second. So hopefully that will update. Yep, I'm going to stop sharing that screen. So I'll suddenly be, it'll become quite big, let's say. So hopefully now you can see my hand down here, and I'm not actually sharing my other things. So what I'll do is I'm going to take a sample, and I'm going to, first of all, this is quite a neat little potentiostat. If I want to put an electrode on there, I'll just take one of these off the panel here, put it on here, snap it. That's the first thing. That's how I make electrical connection with that. In fact, just snap that together and it's done. You can even, so it's very easy. There's no slots. Slots are terrible because slots actually mean that if you've ever used a potentiostat with a slot on it, what you're going to find out very quickly is that it's a great place to get liquid inside the machine. And once you've got liquid inside a potentiostat, that is not a fun place to be. With this, there's no risk of getting the liquid inside. Why should you care about electrochemistry? Look at this. This is quite a large sample, but I'm putting 50 microliters on here. There we are. Doesn't require that much sample. It's a great little science for that. This phone is in Bluetooth connection with this. I'm just going to check the file name here. The reason I'm checking some file names on my phone is so I can find the data later on. I've put the sample on. I'll go to assay and I'll start measurement. So there's a little progress bar flicking around on my phone. There's also a flashing light on the front of the potentiostat, and the flashing light is kind of telling me that the potentiostat is running. I'm just going to here as well. So I'm just going to share something on my screen in a minute. Something, if you follow any of our webinars, you can learn with ZP over time, but actually we always make our assays quick. The reason that we make our assays quick, now at the front of this potentiostat now is actually giving me a green LED, which tells me that it's actually complete the run. It's giving me a result on my phone. And what I'm going to do is I'm just going to change screens with you for a second here. It's what it's done is it's given me an answer on my phone. So we actually ran that progress bar, and I didn't really explain it during the time. That progress bar that was running was actually running a cyclovoltammogram. But when we set up that particular assay, we knew that we were going to have to use this with, I want to say, real people. And real people do not want to see cyclovoltammetry. They are interested in the result. So the result here, for example, is just hold it up, 680 micromolar. They are interested in that result because they want to know what the chemical concentration is, but they certainly do not want to use see the cyclovoltammetry. But the beautiful thing of this is, I'm just going to share I've referenced Julie a few times today. This is Julie. There's Julie .zimmerpeacock.no. I'm inside the data. This is the data that I just recorded. The data went straight to the cloud. This is a real game changer now. This is a potentiostat that didn't require any PC, didn't require any Mac. I ran it off my phone and it immediately put the data into the cloud. I can see the cyclovoltammetry. I can see this is a 2-cycle experiment, first cycle, second cycle, and it actually does the entire data pipeline. It actually processed the data because you've got a whole bunch of tools up here that mean that we can process the first cycle and the second cycle. And on the second cycle, we can do things like filter the data. We can do things like extract values. We can tell it where to find the signal. So I've said here, center on 250 millivolts. What does center on 250 millivolts mean? It means that the software, the cloud software, is actually centered on 250 millivolts. And it's actually, maybe it was actually 300 millivolts. And it was actually looking for the highest peak in there. It's processed the data. I just checked what I've written here. It's actually processed the data. And using this formula, it's calculated from peak height what that chemical composition is. And that's the thing. Most scientific software and scientific papers end with a calibration curve and raw data. This cloud system gives you the cyclovoltammetry in this particular case, but it also can process the data and send the data back to the cloud, and bang, when it does that, now you've got yourself a product, something you can actually, take out of the lab and start giving to, again, that term, real people, but people who are not necessarily electrochemists. So I'm just going to do a quick summary at this point. So I do appreciate the most valuable asset you have actually is your time. So the fact that you've given me your time for the last sort of 51 minutes is appreciated. So I just want to do a quick summary. If you're interested in wearables, we will do a bit more of a deep dive on wearables. And I promise you there's some really good content on that, such as on the 31st of July, one in the morning, one in the afternoon. The one in the afternoon works really well for California. Screen printed electrodes. There are lots of screen printed electrodes in the world. You may even be making some in your own labs. The screen printed electrodes and the irreproducibility of screen printed electrodes is a big holdback on electroanalytical science. It is a real, let's say, problem. I've shown you this afternoon a cloud-enabled potentiostat. This cloud-enabled potentiostat is the difference between TRL3, where most science finishes today, and TRL higher levels, where actually you can start taking this out and actually starting to commercialize it. And you can see at ZPC, we have an entire philosophy that it doesn't end with the potentiostat or the screen printed electrode. It actually ends when you're on the market. And that is a very different philosophy. We hope it plays a role in research and education, but we also hope it plays a role in actual commercialization as well. So I do super appreciate your time. I will be sending you a personal recording of this shortly. If you've got any questions and you're watching this later on YouTube, don't hesitate to reach out to us. But thank you so much for your attention this afternoon, or rather this morning. Have a good day. Okay, take care. Bye-bye.